Coming to the anatomy of testis. What are the internal parts and associated tissues in the testis? We know that testis is present as pairs. It is located in the above the outside the abdominal cavity, and each testis is divided into about 250 compartments called testicular lobules. You can see the testis. This is the cross section of the testis. We can see it is divided into about 250 such compartments, which is known as the testicular lobules. Inside the testicular lobules, we can see 2 to 3 highly coiled convoluted tubules are present. That is known as the seminiferous tubules. So inside the testicular lobule, highly coiled tubular structures that are called seminiferous tubules. The seminiferous tubules at reach to the internal part, you can see it is straight, and this is the straight seminiferous tubules. The seminiferous tubules from each testicular lobule is released out, move out, and it forms a network of tubular system. It forms a network of tubular system, and that is known as the rete testis. Rete testis. And the rete testis leaves from the testis outside through efferent ductules, or this is known as the vasa efferentia. Then this vasa efferentia joins to form the epididymis. This is the head portion of epididymis, and this is the body of epididymis, and the epididymis that continues to form a coiled structure here, that's known as the vas deferens, ductus deferens. Okay, this much I hope that it is clear to you. Now let us see this part, the testicular lobules, once again. The testicular lobules, I told that it is about 250 compartments, and it consists of highly coiled tubule called seminiferous tubule. If you take a cross section of this seminiferous tubule, we can see there are two types of cells are present inside the seminiferous tubule, and between two seminiferous tubule, that is the gap or the interstitial space. In the seminiferous tubule, we can see two types of cells. Two types of cells are present, and that is known as the Sertoli cells and germ cells. Inside the seminiferous tubule, you can see two types of cells. Sertoli cells, it provides nutrition to the developing spermatozoa. It also regulates spermatogenesis by releasing a factor which is known as the inhibin. Inhibin check the overactivity of follicle stimulating hormone, inhibin. So these two are associated with Sertoli cells, providing nutrition to the spermatozoa and releasing inhibin. And another type of cells that present in the seminiferous tubule, that are germ cells. Germ cells are spermatogonia, which undergo meiotic divisions, and finally it forms into the spermatozoa or sperm. Outside the seminiferous tubule is the interstitial space. It consists of interstitial cells that are called Leydig cells. Leydig cells. The interstitial space which consists of Leydig cells and blood capillaries. The function of Leydig cells is to synthesize and secrete male sex hormones, androgens. Among the androgens, we know that the testosterone is the major one. Let us see the anatomy of that seminiferous tubule, the cross section of seminiferous tubule. You can see all these are seminiferous tubules. 1, 2, 3, 4 adjacent seminiferous tubules are seen, and these are the interstitial space, the space between the seminiferous tubule. Inside the seminiferous tubule as well as the interstitial space, certain type of cells are present that we have already learned. Inside the seminiferous tubule, this elongated one, that is the Sertoli cells. Sertoli cells we already discussed. The function is to provide nutrition to the developing sperm as well as releasing inhibin. The interstitial space or the interstitial cells space consists of these cells that are called Leydig cells. Leydig cells are concerned with the production of male sex hormones. We can see the blood capillaries also in the interstitial space. And the germ cells, which include spermatogonium, spermatocyte, spermatids, spermatozoa, like that. Different stages of development of the cells you can see within the seminiferous tubule, altogether called the germ cells. And the connective tissues are also present within the interstitial space. So this is the anatomy of the seminiferous tubule. Initially, I told that the male reproductive system consists of primary sex organs, the duct system, glands, and external genitalia, out of which the primary sex organ testis, somewhat we discussed. Now within that, next we have to discuss the accessory duct system. The accessory duct system, which include rete testis, vasa efferentia, epididymis, and vas deferens. All these seminiferous tubules from the testicular tubules unite to form a straight tubule, which is known as the tubuli recti. You can see from the testicular lobule, diagrammatic sketch, you can see two testicular lobes are always shown here. The highly coiled seminiferous tubule that leaves from the seminiferous tubule, and that portion is known as the tubuli recti. The tubuli recti of all seminiferous tubule or the testicular lobe unite and it forms the rete testis, a network of tubular structure. It's known as the rete testis. And such tubules that leaves from the rete testis, outside leaves from the testis, and this portion is known as vasa efferentia or efferent ductules. The vasa efferentia, its function is to conduct spermatozoa out of the testis. From testis, it is sending out. The sperms are sent out from the testis, that is through the vasa efferentia. And next portion is the epididymis. Vasa efferentia continue to form the epididymis. This is the head of the epididymis and the body of epididymis. And the tubular portion that forms from the epididymis or continues to form the vas deferens or sperm duct. The function of the epididymis is the temporary storage of sperms. The sperms are temporarily stored in the epididymis, and the epididymis continue to form the vas deferens or sperm duct. The vas deferens ascends to the abdomen and it loops over the urinary bladder. It receives duct from the seminal vesicles. This part I already discussed. You can see that the sperm duct or vas deferens that leaves from the testis loop around the urinary bladder and it receives duct from seminiferous tubule. The seminal vesicle. It receives ducts from the seminal vesicle, and the duct from the urinary bladder, which is known as the urethral meatus. Into that urethral meatus, first the duct of seminal vesicles are joining and vas deferens. Along with vas deferens, the duct from the seminal vesicles are also joining into the urethra. This portion is known as the ejaculatory duct. This is a view from backside of the urinary bladder. Okay, is it clear to you? I hope it's clear to you. The duct which leaves from the testis is the sperm duct or vas deferens. From both the sides are united here and it is opening into the urethra. Before that, the vas deferens receives a pair of duct from the seminal vesicle, and that portion is known as the ejaculatory duct. It also receives a duct from the unpaired prostate gland, followed by the bulbourethral gland or the corpus gland. Then it continues as the urethra, which opens exteriorly. We'll see the summary. Vas deferens ascends to the abdomen, loops over the urinary bladder. It receives duct from the seminal vesicle and open into the urethra as ejaculatory duct. Ejaculatory duct, its function is to store and transport sperms from the testis to outside. The urethra originates actually from the urinary bladder. It extends through the penis, which is known as the urethral meatus. The urethra is also known as urinogenital duct because it commonly carries the secretions from the reproductive system and also the secretion, that is the urine, from the excretory system. Penis is considered as the external genitalia of male. It is characterized by a special type of muscle tissues which is known as the erectile tissues, which helps to facilitate insemination. The enlarged end of this penis is known as glans penis, which is protected by or covered by a loose fold of skin, which is known as foreskin or prepuce. Coming to the next is accessory gland of male reproductive system. What are the accessory glands of male reproductive system? There are mainly three: two paired glands and an unpaired gland. A pair of seminal vesicle which secrete a fluid called seminal fluid. Second, prostate gland, which is an unpaired gland. It consists of about 30 to 40 tubuloalveolar glands. It produces some alkaline secretions. That is why the entire secretions of all these glands are basic in nature. The third gland is bulbourethral gland or Cowper's gland. It produces a viscous secretion. I see that the prostate secretions gives a milky appearance, whereas the secretions from the seminal vesicle and bulbourethral gland gives a sticky appearance or sticky consistency. The secretions of seminal vesicle, prostate gland and Cowper's gland are called seminal plasma. All the secretions of these three glands together known as seminal plasma. It is rich in fructose, calcium and certain enzymes. The mixture of seminal plasma and sperm is known as semen, or we can say that seminal plasma plus sperm is known as semen. Now see some characteristics of the semen. The average volume of semen per ejaculation is about 2 to 2.5 ml. The sperm count in each ejaculation may be 200 to 300 million. Semen is an alkaline fluid and its pH is 7.2 to 7.7. It provides the transportation medium and nutrition to the sperms. It also neutralizes the acidic medium of the male urethra and the vagina of female. And this is a pathway of the spermatozoa is summarized like a flowchart here. The sperms that are formed in the seminiferous tubule. From seminiferous tubule it moves to rete testis. From rete testis to vasa efferentia. From vasa efferentia to epididymis. From epididymis to vas deferens. From vas deferens to ejaculatory duct. And from ejaculatory duct to urethra. That's about the male reproductive system of human. And let's see with the female reproductive system in the next class. Thank you.